Hello, everybody. I'm calling in from San Francisco. I'm a virologist here. I direct the Gladstone Institute of Virology. I'm also a professor at UCSF, and I'm part of the UCSF QCRG AViD program. In fact, I'm leading the in vitro virology core for this activity. And today I want to tell you a little bit about what tools we have generated to support the AViD mission and program, and also how these have evolved basically out of our interest in studies for the different SARS-CoV-2 variants. So like many of you, we have in 2020 pivoted part of our research effort to the new virus at that time, SARS-CoV-2. We built specific biosafety laboratory capacity for studying it. And this was actually right after opening of our new facility that is now at the center of the UCSF AViD virology testing. De facto, what we're doing here is testing the SARS-CoV-2 compounds that are coming from the UCSF effort. And we are the first triaging place where we look with a new set of reporter viruses for activity, and then we send it out to the rest of the virology core, which is actually situated in Paris, New York, and in London, and Texas, sorry. So one of the reasons why we thought we could contribute was that we had actually a whole wide range of human cell models at the time in 2020 growing, including some lung organoids that we had established for influenza work, but also intestinal organoids and iPSC-derived cardiomyocytes. And together with many colleagues at UCSF, but also around the country, we have actually tested infection very early on in many of these cell types. And you see on the bottom here, actually in green, the double-stranded RNA centers that mark the infection in these cell types. Specifically, the cardiomyocytes become sensitive, very sensitive to the viral replication and have unique pathology in response to it. But the different experiences in the cell models now is also coming very handy for the analysis of the different compounds together with our colleagues and together with the molecular clones that we have been generating. This is really at the heart of the cell culture and the in vitro virology core. We also have established some in vivo models and contribute to the in vivo core, which is led by Adolfo Garcia Sastre at Mount Sinai and Chris White, actually. So we have, and we support some of their efforts with the models that we have established, which is the classical mouse models here for SARS-CoV-2, and recently also the hamster model. So today I want to focus on two tools that we have developed together with colleagues here at UCSF. One are virus-like particles. They are slightly different than the usual pseudoviruses or pseudotype viruses that are being used. And are more authentic. And the second one is actually the assembly and the generation of full-length SARS-CoV-2 genomes. And Tiago has given an excellent introduction into the genome, so I don't have to go very much into detail. Suffice it to say that the genome is very long, and it's not super easy to assemble it rapidly and correctly. And we have, I think, some contributions here that have helped to not only find new biology, but also to construct new genomes. And we are, this is published, and this has been also shared with many colleagues already around the country. So the variants that triggered these the development of these tools were initially Delta. and later the Omicron BA.1 variant. And I'm going to talk a little bit about these two variants and how we analyze them with the tools we generated. So the first was a rapid assessment of SARS-CoV-2 evolved variants using virus-like particles. And here, as I said, we're focusing on Delta. This is a close collaboration with Jennifer Doudna's lab, especially a very talented postdoc in her lab, Abdullah Sayed, together with a postdoc in my lab, at the time a postdoc, now a research investigator, Taha, and Takaku Tabata, who was actually technically supporting the effort. And the idea was really that particles, SARS-CoV-2 particles, are very unique, composed out of 4 structural proteins, the S, the M, the E, and the N protein. And the most easy way to study the particles, and especially the spike protein, the S protein that is exposed on the surface, is really to initially make some pseudoviruses, which are either HIV-based, lentivirally based, or VSV-based, and that has led to very many very interesting and very important early studies of especially also the immunogenicity, but also the capability of the spike proteins to mediate entry into the target cells. However, they don't look authentically like the SARS-CoV-2 genome. They're basically another viral capsid, and then decorated with the glycoprotein of SARS-CoV-2. In order to really find something that is more authentic, that helps with all the studies that I have just mentioned, immunogenicity of a spike and entry of these particles, but also can help with studying the assembly of these particles in the cell, because it really is the authentic virus-like particle. We set out to really develop a system where we could not put the spike simply onto another virus, but where we really assemble a capsid, package the RNA into an authentic capsid of SARS-CoV-2, and then decorate it with the spike protein. To do this, it was really important to identify the packaging signal for SARS-CoV-2, because usually this is very much dependent on the capsid. And if you use a foreign capsid, you can use the packaging signal that is actually known. If you don't know it and you want to build an authentic one, you have to really find out how SARS-CoV-2 actually gets messages and genomes into these particles. This was done with this was mapped by Sayed, and he used information that was available from SARS-CoV-1 and mapped it down to these 20 amino acids at the junction of the ORF1ab and the S genes here. To test whether it was correct, he basically hooked it up to a luciferase reporter and did a co-transfection assay where he basically expressed all 4 proteins together with the reporter, including the new packaging signal, transfected it into cells, harvested the supernatant, and then tested in target cells that expressed the relevant receptors that whether there is really infection here. And you can see that these experiments only work when you have a very carefully calibrated expression system of these plasmids, but also only if you have the 4 together. If you lack any of the individual ones, you don't get any signal in the target cell. So that now allowed us to not only study mutation that have evolved in the spike protein, but also focus on other proteins. And one protein that was particularly interesting to us was the N protein. I told you that was at the start of the Delta wave. And there was actually quite a few mutations that have evolved in this N protein. And N is a very important protein. It is sort of the capsid that is composed out of practically two domains: the N-terminal domain that is doing the RNA binding together with the C-terminal domain, because that's basically the main function, to incorporate the RNA. but also the oligomerization is very important because that really forms the capsid, and that is endowed in the C-terminal domain. These two domains are connected by this serine-arginine-rich motif, which I will come back to in a minute, because that is actually the motif where we find a lot of the mutations that have been accumulated up to that time in the pandemic, highlighting especially these 4 mutations, because when we tested now all of these mutations individually in the system that I just told you, by just using the N protein in our co-transfection assay, including these individual single mutations into it, we could see that these 4 mutations in the linker domain were actually increasing the signal that we get in the target cell, indicating that there was either more particles made, or more infectious, or more better infectious particles made, or better entry. And so to differentiate between that, we actually isolated the particles from the supernatant after the transfection, and then analyzed them by Western blot and by Northern blot. And by Western blot, we saw that there was really not anything that, when you focus on these 3 mutations here, which are actually the ones that increase the activity, nothing was really standing out that was unifying these 3 mutations, only when we started to look at the RNA. So what was happening is that these 3 mutations really increased the incorporation of the RNA into the particles, indicating that the variant has probably evolved to increase RNA incorporation here, or to optimize it. We have at that time used the infectious clone system that was generously and provided by Pyongyang at UTMB, and have actually shown that 2 of these mutations here that we tested, indeed, if you just put 1 single mutation in the N protein into, you can recapitulate the increase in particle production and in replication here. So the model here is that to study either, one of the 4 or mutations in one of the 4 proteins that are making up an authentic SARS-CoV-2 particle, you can use that VLP system. We have used it to study mutations in the Delta variant, and have found that specifically the mutations in the linker region are very effective to actually increase assembly of these particles, and especially increase incorporation of the RNA. We're using this. The advantage of that VLP system is that it is not used in BSL-3. It's a BSL-2 system, and it's very effectively used to currently to study anything in terms of drugs that is that is affecting assembly of the particle. And this is used as a pre-screening before we go and test these drugs in the infectious clone system. Syed has also worked very much on this model, and has a second paper almost accepted, where he's actually showing that the linker region and these mutations are changing the phosphorylation status of the N protein, and that actually changes the way how N functions either as a capsid in particle production, or as a supporter of RNA replication in the cells. So with this, I turn to the second part of my talk, where I now talk about full-length SARS-CoV-2 genomes. And the Omicron variant. So here, this is led and actually also conceived by Taha, really the major driving in generating this molecular reverse genetic system, together with a student at this time and now graduated PhD, Irene Chen. What was really triggering our interest here in getting into this tool development was the emergence of Omicron BA.1 at the time, and some studies that we and others did in cell cultures, but also in mice, where we showed that the Omicron was very much attenuated in the way that it caused much less disease. We had no weight loss and also no death in these mice. And we had, importantly, up to 3 logs less viral titers in various tissues in the mice. So really indicating that BA.1 was attenuated. And that was, of course, widely studied with the many mutations that have evolved in the spike protein, including in our study that I just mentioned in the mouse, but also Sayed did a beautiful study with his VLPs showing that some of the immunogenicity of these spike proteins have really totally changed. But we wanted to focus on the other mutations that have evolved, including mutations that have evolved in the ORF1ab. And you have just heard that this is really what encodes the replicase complex and is important for genome replication in the cell. So it's not really amenable to very easy, pseudotype virus studies, but really requires studies that have either the full-length virus, infectious virus, or has so-called replicons. So in order to now start making a lot of these mutants, we set out to facilitate the existing protocol in order to shorten the time frame and also enhance the success rate that we had. To do this, we actually divided the genome into 10 different plasmids. That was also a little bit aligned to having a better opportunity to mutate individual proteins by or open reading frames by just using one plasmid and not have to work across different plasmids. But it was also designed to have less toxicity of these plasmids in bacteria, which often leads to very low yield. And then instead of ligating these individual steps one by one, we developed a Golden Gate assembly strategy where we assembled everything in one pot with a PBAC plasmid, generating a very large construct that contained the whole, basically the whole genome, that we can then use to either in vitro transcribe and generate RNA or use as a DNA plasmid directly in transfection assays that we use to rescue virus and generate the relevant SARS-CoV-2 variants. That whole process is done very effectively and very rapidly in a few days, maximally a week. And you see that it also has it actually has a very high success rate. Here in the Golden Gate assemblies procedure, you see that that you have really almost 100% assembly in after 30 cycles in the Golden Gate procedure. And when we looked by sequencing, these were mostly up to 100%, but minimally 80% of them were correctly assembled, had no mutations, and were also in the right order and in the right orientation. We tested these recombinant viruses, regardless if they were generated from DNA or RNA in different cell types, but also in the mouse. And we did see that we did not find any difference with the original isolates that we tested here. We also now started to sort of go after mutations that have been generated here or that have evolved in Omicron in the ORF1ab open reading frame. And to do this, we started out to test different Delta cron variants. I want to point out that all of these are existing in nature, and we were very careful to not generate any viruses that were novel. Because Omicron and Delta had a considerable overlap, there's actually quite a bit of recombination between those, and these Delta cron viruses exist, both those that have basically Omicron ORF1AB and Delta structural and ORF proteins here, or vice versa. And there's also one or two viruses where just the spike protein was exchanged between Delta and Omicron. And so using these naturally occurring viruses that we reconstructed, basically, we found that indeed, if we used the Omicron ORF1AB, or if we used the Omicron spike, both of these were basically decreasing the replication of the original Delta strain, indicating that both the mutations in the replicase, but also the mutation in the spike protein are important for replication, for the attenuation of Omicron. And the same was done when we tested, basically, when we compared this with the original Omicron replication pattern, when we used the Delta replicase, or when we included the Delta spike protein, we could see that that increased the replication compared to the Omicron strain here. And a similar result was found in this Nature paper here from the Boston group, who made actually chimeras between Omicron and the original Washington strain. Now, to study now in further detail where the attenuation lies, we did not want to go into any gain of function studies. We did not want to go into making individual mutations in the full-length virus. We actually used a replicon system because that has an intact ORF1AB and replicase, but it is not generating infectious particles, because we take specifically the spike protein out, because we also did not want to have any contribution of the spike protein to our phenotype here, but wanted to really focus on the replicase phenotype here. We replaced these with a couple of reporters, and we're using these viruses either full length or as a replicon very successfully for checking our compounds here in our core facility. We do these replicons, we assemble them exactly the same way like the infectious viruses, only because we're lacking spike, we have to co-transfect the spike in trans. We can then basically generate single round infectious particles that we can then use to infect target cells, and then study either the entry or the replication of these viruses. And what you will see here, what we test is a lot of stuff after just transfection. We'll just see how you transfect this replicon into a cell line, and then see what is the autonomous replication or the luciferase value here. Or we testing it after a single round infection, which is then basically augmenting the phenotype that you have in the original cell, and you get it in the target cells also. And you will see that we have overlapping results in both approaches. So, but when we just compare a Delta and an Omicron replicon, which now doesn't have any more spike, you can see that the pure replication of Omicron is decreased, both after transfection and after a single round infection. We see basically the same decrease, supporting the model that these, not only the spike mutations, but also mutations in the replicase are very important for the attenuation. And now in this replicase, in this replicon system, we have gone and exchanged each individual protein that had a mutation between a difference between Omicron and Delta. And we used the Omicron background and replaced it and cloned into it the Delta proteins that had a mutate that were different from the new Omicron strain. And we could see immediately that one protein stood out, which was the NSP6 protein, which almost completely restored the activity of the Delta replicon if we just used the Delta NSP6. Interestingly, the opposite was true for the NSP5 protein, because here we see that that replacement of the Delta protein is actually decreasing the Omicron activity, indicating that NSP5 has gone through an optimization process in the Omicron virus, interestingly, if we replace them both together, the NSP6 phenotype is the dominant one. And that's why we have focused mostly on the NSP6 protein. It is a very interesting protein. It was at the time just reported. It is important for the membranous web or membrane proliferation around the replicon, the replicase complexes, the RNA replication centers in the infected cell. And it was just reported that it was actually important for recruiting lipid droplets, which are energy carriers, but also, lipid carriers for potentially membrane generation. That these were important, that NSP6 was an important factor to recruit and consume these lipid droplets in infected cells. So to test whether there was a difference between the Delta NSP6 and the Omicron NSP6, we just expressed the proteins in cells and then stained either for the protein in green or for red for the lipid droplets. And you can see that in the Delta, we really don't have much red, which means that the Delta NSP6 is very effective in consuming the lipid droplets, while in the Omicron NSP6, we see a lot of lipid droplets left, similarly to the untransfected cells. But because the Omicron NSP6 is obviously less effective in this function to recruit and consume the lipid droplets, and that is quantified here. And we did the same thing with the replicons, where we not express the protein alone, but in the presence of the replicating RNA genomes. And you can see that there's relatively little if you use Delta. There's relatively lot of the red if you use the Omicron. And then if you use the Delta and just replace the NSP6 from Omicron, you see that it resembles the Omicron phenotype, and vice versa. If you use an Omicron replicon and you replace the Delta NSP6, you can see that it diminishes the lipid droplets that are left in the cells, indicating that indeed the Delta NSP6 is good at the function of consuming these lipid droplets and presumably helping the membranous web generation here, while the Omicron NSP6 is not as efficient in doing this. And so what we think we have shown with these different models is that there is actually a multi-step attenuation for the Omicron variant. Apart from spike, we also think that there's a significant amount of attenuation in the RNA replication. And we think that specifically the generation and the conformation and the maintenance of these double membrane vesicles, or the membranous web that is protecting the replicase complex, is actually very important and is an important rheostat to dial up and down in the viral fitness for different variants. And so what we're doing is we're using this technology that I just described to you by generating various reporter viruses and replicons. Again, the replicon is a not BSL-3 technology and can be adapted to BSL-2 or BSL-2 plus. And we're using this a lot for testing, inhibitors, various inhibitors of the different, replicase and protease components. We have also used it to actually generate systematically mutations in all of the proteins that have enzymatic activity. One of them is NSP3. And we have this paper, actually Taha has this paper almost accepted, where he showed a single point mutation that kills enzymatic activity and how it replicates in cells and in mice. But we're also very interested in this role of NSP6 in viral RNA replication. I think we're working very hard on this. I think, as I said, it's really pointing to these to the architecture of the replicase complex in the cytoplasm as an important rheostat where the variant evolution is taking place apart from spike. And we're really interested in the lipid droplet part of it, because we have a long-standing interest. I'm coming back a little bit to hepatitis C, like Tiago did at the beginning of his talk. We have been working, a lot on the molecular biology of hepatitis C, and specifically the role of lipid droplets in this viral replication, and had found that lipid droplets are important for assembly, but also for RNA replication, similar to what we see now, that lipid droplets are a very important part of this membranous web that is protecting the RNA replication complexes. So we're very interested to see how that applies to SARS-CoV-2 and what are the details here with the various variants. So with this, I stop. Thank my really awesome group. We're just coming back from a lab retreat. We had a lot of fun. I mentioned Taha and Irene, but many others here in the crowd have actually contributed to many of these studies. Really close collaborators, especially Jennifer. Jennifer has a major effort in Berkeley, but also has a lab at the Gladstone Institute right next to mine, and that has led to a lot of really interesting collaborations and wonderful exchanges. Want to especially thank Nevin, the leader of the QCRG AViD program, many and NIH for supporting the work that we're now doing with these tools to generate new or to test new compounds. Many of the supporters that we had that helped with our SARS-CoV-2 work, and specifically all the colleagues in the UCSF QCRG AViD program with whom we all interact very closely, as we are the first step for testing basically the compounds in viruses. With this, I thank you very much and look forward for any questions.